Hello students, welcome back to a new video of Sunanda's tutorial. It is the part 3 video of your chapter 5, which is molecular basis of your inheritance. So it is the part 3, and in this video we will discuss about the human genome project and DNA fingerprinting. So what is human genome project? Human Genome Project or HGP was an international scientific research project which was completed in the year 2003. It takes total 13 years to complete or to sequence the whole human genome, which is consist of 3.3 billion base pairs. 3.3 billion base pairs. So it takes 13 years to complete this project from 1990 to 2003. That means 13 years. In these 13 years, they sequenced the whole human genome. The Human Genome Project led to the growth of bioinformatics, which is a vast field of research. The successful sequencing of human genome could solve the mystery of many disorders in human and gave us a way to cope up with them. Then the goals of human, what are the objective or goal of Human Genome Project? Optimization of data analysis, sequence the whole genome of human, then identification of complete human genome, creating human genome sequence database and to store the data, then taking care of legal, ethical and social issues that the project may solve. Then method of Human Genome Project, these are the goal, and then the methodologies. The complete gene set, in this project, two different and significant methods are typically used. First one is expressed sequence tags, wherein the genes were differentiated into the ones forming a part of the genome and the other which express RNAs. Then second, sequence annotation, wherein the entire genome was first sequenced and functional tags were assigned later. Then the process of Human Genome Project. The process, the complete gene set was isolated from a cell, then split into smaller fragments. Then the DNA structures was amplified with the help of vector, which is mostly bacterial artificial chromosome and yeast artificial chromosome. Then smaller fragments then sequenced using DNA sequencer. On the basis of overlapping regions, sequences were then arranged. All the information of genome sequence was then stored in a computer-based program. In this way, the entire genome was sequenced and stored as genome database in computers. So the genome mapping was the next goal, was achieved with the help of microsatellite, which is repetitive DNA sequence. So it is the total or overall process of Human Genome Project. Then the salient features of human genome. Human genome contains 3164.7 million base pairs. Average gene consists of 3000 bases, but sizes vary greatly, with largest known human gene being dystrophin at 2.4 million bases. The total number of genes is estimated at 30,000, much lower than the previous estimates of 80,000 to 1,40,000 genes. Almost all nucleotide bases are exactly the same in all people. Functions are unknown for over 50% of discovered gene. Less than 2% of genome codes for protein. Repeated sequences make up very large portion of human genome. Repetitive sequence are stretches of DNA sequence that are repeated many times, sometimes 100 to 1000 times. They are thought to have no direct coding function, but they shed light on chromosome structure, dynamics, and evolution. Chromosome 1 has most genes, that is 2968 genes, and Y chromosome, which generally the male have Y chromosome, and that Y chromosome has fewest, that is 231 genes. Scientists have identified about 1.4 million locations where single base DNA or single nucleotide polymorphism occur in human. This information promises to revolutionize the process of finding chromosomal location for disease associated sequences and tracing human history. Then what are the application of this human genome project and future challenges? So as the goal of human genome project were achieved, it led to great advancement in research. Today any disease arises due to some alternation in certain gene, it could be traced and compared to the genome database that we are already have. In this way, a more rational step could be taken to deal with the problem and can be fixed with more ease. This project this project has opened up new horizons which can be learned in much detail with our expert faculties. So this past research or human genome project or study about the sequencing the whole genome of human have many applications. They helped us a lot, and they can study all the gene in a genome, for example, or all the transcripts in a particular tissue or organ or tumor, or how tens of thousands of gene and proteins work together in interconnected network to orchestrate the chemistry of life. So it is all about human genome project. Next, what is DNA fingerprinting? DNA fingerprinting is a technique that shows the genetic makeup of living things. It is a method of finding the difference between satellite DNA regions in the genome. And 99% of a base sequence among humans is same. So it is just little bit difference among all of us. It is these differences in sequence of DNA which make every individual unique in their phenotypic appearance. If one aims to find out genetic differences between two individuals or among individuals of a population, sequencing the DNA every time would be a daunting and expensive task. Imagine trying to compare two sets of 3 into 10 to the power 6 base pairs. DNA fingerprinting is a very quick way to compare the DNA sequence of any two individuals. DNA fingerprinting involves identifying the differences in some specific region in the DNA called repetitive DNA. Because in this sequence a small stretch of DNA is repeated many times, these repetitive DNA are separated from the bulk genomic DNA as different peaks during density gradient centrifugation. The bulk DNA forms a major peak and other small peak are referred to as satellite DNA. Depending on the base composition, AT rich or GC rich, length of segment, number of repetitive units, satellite DNA is classified into many categories such as microsatellites, mini satellites, etc. These sequences normally do not code for any proteins, but they form a large portion of human genome. This sequence show high degree of polymorphism and form the basis of DNA fingerprinting. Since DNA form from every tissue from an individual, So same degree of polymorphism, they become very useful identification tool in forensic application. Further, as polymorphism are inheritable from parents to children, DNA fingerprinting is the basis of paternity testing in case of disputes. As polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as DNA fingerprinting, it is essential that we understand that DNA polymorphism means in simple terms. Polymorphism means basically the variation arises due to mutation. In chapter 4 you know what is mutation. New mutation may arise in an individual either in somatic cell or in germ cell. If germ cell mutation does not seriously impair individual's ability who can transmit the mutation, it can spread to other member of population. Allelic sequence variation has traditionally described as DNA polymorphism. If more than one variant at a locus occur in human population with frequency greater than 0.01, in simple terms, if an inheritable mutation is observed in a population at higher frequency, it is referred to as DNA polymorphism. The probability of such variation to be observed in non-coding DNA sequence would be higher, as mutations in these sequences may not have any immediate effect or impact in an individual's reproductive ability. These mutations keep on accumulating generation after generation and from one basis of variability or polymorphism. There is a variety of different types of polymorphisms ranging from single nucleotide change to a very large scale change. For evolution and speciation, such polymorphism play important role. The technique of fingerprinting was initially developed by Alex Jaffery. He used satellite DNA as probe that shows a very high degree of polymorphism called variable number tandem repeats or VNTR. The technique was used as used earlier involved southern blot and hybridization using radio labeled VNTR as a probe. It includes isolation of DNA, then digestion of DNA by restriction endonuclease, then separation of DNA fragment by electrophoresis, then transferring this DNA fragment to a synthetic membrane such as nitrocellulose membrane, then hybridization using labeled VNTR probe. Then detection of hybridized DNA fragment by autobiography. A schematic diagram is there in figure 5.16, which is the schematic representation of DNA fingerprinting. The VNTR belong to the class of satellite DNA referred to as mini satellite. A small DNA sequence is arranged randomly in many copy numbers. The copy number varies from chromosome to chromosome in an individual. The numbers of repeats show very high degree of polymorphism. As a result, size of VNTR varies from 0.1 to 20 kb. Consequently, after hybridization with VNTR probe, autobiogram gives many bands of different size. These bands give a characteristic pattern for an individual DNA. It differs from individual to individual in a population except in case of monozygotic twins. The sensitivity of technique has been increased due to the use of the polymerase chain reaction. Consequently, DNA from single cell is enough to perform the DNA fingerprinting analysis. In addition to application in forensic science, it has much wider applications such as in determining the population and genetic diversity. Currently, many different probes are used to generate DNA fingerprinting, and it is the structure schematic representation of different steps that are involved in DNA fingerprinting. So it is all about your molecular basis of inheritance, chapter 5. In my next video I will upload the chapter 6 of your 12th class NCERT biology. So stay tuned and like my videos, share with other students and your friends, and subscribe my channel Sunanda's tutorial. Thank you.